Good morning, everyone. Today I am going to discuss the process of oogenesis with you. If you talk about oogenesis is basically the process of egg production, and as you all know, it greatly differs from spermatogenesis. Mammalian egg maturation can be seen as having four stages. The first of this stage is stage of proliferation. In this, in human embryo, thousand or so primordial germ cells reach the developing ovary, dividing rapidly from second to the seventh month of gestation. They generate roughly around 7 million oogonia. So this stage is basically taking place before the birth of an individual. That means it is from second to seventh month of gestation. While most of these oogonia die soon afterward, the surviving population under the influence of retinoic acid enter the next step and initiate the first meiotic division. They become primary oocyte. So out of 7 million oogonia which are present in the gonads, in the ovary, only few of them enter the first meiotic division and became primary oocyte. And this first meiotic division does not proceed very far, and primary oocyte remain in the diplotin stage of first meiotic prophase. And this prolonged diplotin stage is sometimes referred to as dictate resting stage. Now the thing is, at birth, all the oocyte that are present inside the gonads, that are present inside the ovary, are arrested at diplotin stage of first meiotic prophase, and they will initiate, or they will develop further only when the individual is going to reach the puberty stage. With the onset of puberty, group of oocytes periodically resumes meiosis, and at that time luteinizing hormone from pituitary gland releases this block, and the oocyte resume meiotic division. So the next is this arrested first meiotic division will resume once an individual reaches puberty, and the resulting secondary oocyte proceed to the second meiotic metaphase and undergo maturation steps. Now this secondary oocyte will be arrested at metaphase stage, and further development will be initiated only if the fertilization takes place. And this maturation of the secondary oocyte within the Graafian follicle involves the crosstalk of paracrine factors between oocyte and its follicle cells, both of which are maturing during this phase. The follicle cells activate the translation of stored oocyte mRNA encoding proteins such as sperm binding proteins that will be used for fertilization, and cyclins that control embryonic cell division. So in this stage, basically secondary oocyte is being prepared for fertilization, because for fertilization egg requires large number of proteins which will be helpful in binding with the sperm. After the secondary oocyte is released from the ovary, meiosis will resume only if fertilization occurs. At fertilization, calcium ions are released in the egg, and these calcium ion release the inhibitory block and allow the haploid nucleus to form. So in further videos, I will be explaining how this calcium ion plays a very important role. When we will doing fertilization, we will see that calcium ions plays a very important role in preventing polyspermy. So this is the summary of what we have done so far. Here you can see we have a oogonia which is diploid. It undergoes mitotic division, and this is taking place before birth. and we have a primary oocyte which is still diploid, and this primary oocyte gets arrested in prophase of meiosis 1 and it remains in this stage for around 12 to 13 years. Then on reaching puberty, secondary oocyte is formed after meiosis 1 and this secondary oocyte is again arrested in metaphase of meiosis 2. Then ovulation, that means release of egg from the Graafian follicle takes place and further development will occur only if this secondary oocyte is fertilized, and a single secondary oocyte is released each month after puberty. Well, this slide shows the difference between two processes, oogenesis and folliculogenesis. I told you last year also that folliculogenesis is basically a sub part of oogenesis. If you see here, follicle development in ovary, we have two terms. We have primordial follicle, primary follicle and primary oocyte. So you should not be confusing between the terms primary follicle and primary oocyte. Primary oocyte is basically the stage of the egg, fine, and primary follicle is the stage of the complete structure, that means egg plus the surrounding follicular cells. So in folliculogenesis it begins with primordial follicle, then we have primary follicle and it extends up to the Graafian follicle, and even after the formation of Graafian follicle, release of secondary oocyte and formation of corpus luteum is to be included in the process of folliculogenesis, whereas oogenesis is far much broader and consists of few additional steps at the beginning as well as at the end. We can see here that development or change of oogonia to next stage is included in oogenesis but not in folliculogenesis. Fine. Then in the last also, after the release of secondary uh, oocyte from the egg and formation of corpus luteum, folliculogenesis is up till the formation of corpus luteum, whereas in oogenesis we also include the fertilization of secondary oocyte with the sperm and completion of meiosis 2. So oogenesis is far much broader as compared to folliculogenesis. This slide shows maturation of the mammalian oocyte. Now, ovulation in mammals follow one of the two patterns depending upon the species. One type of ovulation is that which takes place in rabbits and minks, and they have a reputation of procreative success. And in this type of ovulation, uh, the ovulation is stimulated by the act of copulation. That means physical stimulation of the cervix triggers the release of gonadotrophins from the pituitary, and these gonadotrophins then signal the egg to resume the meiosis and initiate the events that will expel it from the ovary. This mechanism ensures that most copulations will result in fertilized ova, and the animals that utilize this method of ovulation have a reputation of procreative success. The second type of pattern is that takes place in most of the mammals, and we call the ovulatory period as estrus or heat. The environmental cues, most notably amount and type of daylight, stimulate the hypothalamus to release gonadotrophin releasing hormone, and these gonadotrophin releasing hormone stimulates the pituitary to release gonadotrophin follicle stimulating hormone as well as luteinizing hormone that causes the ovarian follicle cells to proliferate and secrete estrogen. Estrogen enters certain neurons and evokes the pattern of mating behavior characteristic of the species. The gonadotrophins also stimulate follicular growth and initiate ovulation. Thus, mating behavior and ovulation occur close together. But in humans, we have a variation. As you know, humans also have a periodic cycle, but that cycle is not regulated by daylight. Fine. We have a periodic cycle which is termed as menstrual cycle, and it entails the periodic shedding of blood and endothelial tissue from the lining of the uterus. This slide shows that menstrual cycle represents the integration of three different cycles. We have ovarian cycle, uterine cycle, and cervical cycle. In ovarian cycle, the function is to mature and release an oocyte, whereas uterine cycle prepares the uterus lining for the fertilization and development of blastocyst. Whereas cervical cycle, it functions allow the sperm to enter the female reproductive tract only at the appropriate time, and all these cycles are regulated and coordinated through the hormones of pituitary, hypothalamus, and ovary.
Now the second important difference between oogenic meiosis and spermatogenic meiosis is that in spermatogenic meiosis, the spindle is formed by centrioles, whereas in case of oogenic meiosis, the spindle is formed by microtubules. Why? Because oogenic meiotic spindle lacks centrioles, because oocytes don't have centrioles. Rather, microtubules of the meiotic spindle are organized by mRNA and enzymes located on the chromosome and spindle fiber themselves. Instead of two centrioles, numerous microtubule organizing centers form around the nuclear envelope at first meiotic prophase, and these spindle then uh, join together once the nuclear envelope breaks down. Now this diagram shows two blocks that occurs in case of amphibians. We have this first block and then we have a second block. See, we have a mitotic arrest, first meiotic prophase. This is arrested and this is termed as diplotin block. And why is this block? Because there is no active MPF. MPF is mitosis promoting factor. Fine. and this block is removed only when progesterone is present. This progesterone helps in secretion of CMOS, and this CMOS basically helps in converting inactive mitosis promoting factor to its active form by phosphorylation. Fine. The phosphorus group add hoकर inactive MPF becomes active MPF, and MPF in turn is form of two subunits. We have P34 and cycline, and these subunits join with each other and with the phosphate group only if CMOS is present, and CMOS will be present or CMOS will be secreted only if progesterone is present. So this block is removed and first meiotic metaphase and then first meiotic division is completed. Now the egg enters into second meiotic metaphase, and this is again arrested at this. Now you can see this block here, fine, and this block will be uh, crossed only if CSF, cytostatic factor, is degraded, because CSF is basically blocking the egg from crossing this barrier, and so the egg remains in second meiotic metaphase. And how this CSF is degraded? It is because of fertilization. Once the egg is fertilized, influx of calcium takes place, and this calcium then binds with colmogylin protein, and this colmogylin protein in turn activates two proteins, campk2 and calpin2, and both these proteins in turn help in degrading CSF, and which will ultimately release this block, and meiosis 2 will be completed. So this is the slide which shows how basically uh, nerve cells and oocyte is derived in insects from a single oogonia. You can see this is a single oogonia. It divides into two, then again it divides, then it divides into four, ultimately it divides into 16 cells. Now the thing you should notice here is you can see this red structure which is labeled as fusome. This fusome don't divide, it moves only to single cell. Like in this case, two cell stage, this fusome will migrate to cell number 1, and then again in 4 cell stage this fusome is retained by cell number 1. Ultimately what happens when the 16 cells are formed, the fusome is present only in cell number 1. All the other 15 cells don't have fusome, but you can see that all these cells retain connection with the cell number 1. Why? Because it has fusome. So basically that cell which has the fusome is destined to become the oocyte. Rest all other cells will become nerve cells, and they are termed as cytocytes. Fine, this is oocyte and others are cytocytes. So basically what happens, the fusome is retained by only one cell, and that cell is termed as oocyte. That cell will become the egg. Rest all the 15 cells are going to function as nerve cells, and they will have collagen chromosomes. They will undergo active transcription, and they will form large amount of RNA, which will ultimately be transferred to this cell number 1, uh, which needs these RNAs for uh, functioning after the fertilization.